Alright, so we're going to dive into our first category of biomolecules here. So we're going to talk about nucleic acids. And so remember, these are for genetic information storage. So this is like the blueprints of the whole operation. And we'll talk about proteins later, and that's like the machinery and a couple different ways of storing energy. But this is the one, there's going to be some important details here in terms of keeping the DNA from getting manipulated and damaged. So protecting the DNA, being able to detect a mismatch of some kind, something that's awry, and being able to fix it is a very important detail. So if you fail to do that in a cell, it can undergo cell death. It can become cancerous. There's a number of things that can happen. Maybe it continues to function and live, but not as efficiently. Or maybe through some random mutation it actually turns out better. But more often than not, it's going to lead towards cell death and cancer. Things are pretty well optimized at this point. So you've really just got to protect the DNA. So there's a reason it's in the middle of the cell. It's in the nucleus of the cell. That other nucleus, the one that I never talk about till now, and I probably won't ever talk about again. It's to keep anything from getting to it. So something's got to get into the cell, and then it's got to get into that organelle. It's got to get into the lockbox inside the cell to access the DNA. So it's a tightly controlled area. Now, what is DNA made of? Well, nucleic acids. And I'm going to talk more about the structure. So it isn't just these molecules. So I show it going to a backbone here. So DNA has a sugar phosphate backbone. RNA has a sugar phosphate backbone. I'm going to get to that later. And you're going to see the same trend when I talk about all of these biomolecules. I'm going to start small. We're just going to get bigger and bigger and bigger until we're talking about the big macromolecules. So this is long-term genetic storage, information storage, I should say, genetic information storage. And RNA can just, it's the cheap copy. We make a cheap copy. That gets sent out of the nucleus. That gets sent out into the rest of the cell. So if something happens, if the RNA gets torn up, whatever, you still got the master copy. You just make another RNA copy and you send it out there. That's what you do. So these are the nucleic acids you find in DNA. And so you might remember that you have a GC base pairing, if you've covered that in biology yet. And this is what it looks like. And you don't have to memorize these structures, let me be clear. But you might want to remember what a nucleic acid looks like, so be able to recognize it when you see it. So you just see these kind of rings with some nitrogens in them all over the place. You got yourself a nucleic acid. Like you got a ring with nitrogen. Now it might be a single ring or something with a double ring. Same thing here. We've got a single ring, something with a double ring. We'll talk more about that in a second. That's what a nucleic acid looks like. That's the key feature. Carbohydrates are definitely not going to look like that. Proteins are, there's a little bit of nitrogen in proteins or in amino acids, so you got to be careful there, but you're not going to see these ring structures. So looking for rings with nitrogens all over the place. You see that? Nucleic acid. All right. So what is this base pairing? I always like to talk about this on a chemical level. So this is one of the ways that DNA is protecting itself, or is being protected from, you can think of it like data corruption. And so how this base pairing works is you get hydrogen bonds. So these double bonded oxygens, these ketone groups, can actually participate in hydrogen bonding. Now they don't have the ability to send a hydrogen bond. They don't have an H to send out, but they have the part of an oxygen, namely the lone pairs. So remember, it has the lone pairs here. Those can receive a hydrogen bond. So this is like a half hydrogen bonder. So you've got like a key, and you've got a lock that can fit in a key. You can't unlock a lock with another lock cylinder, but you can fit a key in there. And so this can send hydrogen bonds, and this nitrogen can also receive them. Remember, these NH groups can send and receive, just like OH groups can send and receive. You'll notice that DNA is going to solely rely on nitrogens sending hydrogen bonds. And they might receive them, but you also get some double bond oxygens that receive them. But only NH groups. There's no OH groups here. So just keep that in mind. So I did these dashed lines just to show. So we have 3 areas of hydrogen bonding. I didn't draw these, exactly how they would be in 3D space. So just be aware that, there's these 3 spots. And when we look at adenine and thymine, things line up so that we get some hydrogen bonding as well. And, oh, that's what was missing. I was missing an H there. Okay, so we get some hydrogen bonding here. So this nitrogen needs to make 3 bonds. Where did the third one go? And it went to the hydrogen. All right, and we get some hydrogen bonding here. So in this case, there's only 2 locations for hydrogen bonding. And this is where we start talking about kind of the data fidelity, maintaining the DNA. So if I have an adenine here, it should be paired with a thymine. So DNA is going to have 2 chains of nucleotides. I'll talk about those more. So that's going to be everything. So we have the backbone and the nucleic acids. But essentially you have these 2 polymer chains, and they're complementary to each other. So if I have, say a cytosine on this chain, then across from it, I will have a guanine. They'll pair. And then what holds these 2 chains together, they're not covalently bonded. It's like a giant zipper. I can take them apart, but I have to undo a bunch of hydrogen bonds. There are, potentially hundreds of thousands or millions of hydrogen bonds that hold these 2 things together. And lots of small interactions can add up to be a very strong force, and that's what you get with DNA. But what you can do, and you'll see this in your biology class, is you can open a work site. So you can open it up, and then you can create a spot where you can get some proteins in there, and you can start doing DNA replication. You can do transcription, where you make RNA. So DNA replication would be for cells, and what that does is you end up with 2 chains. Now, you don't need to know this for my class, but I always like to help you guys out with biology. Once upon a time, I was a biochemist. So it was a dark time in my life. I've worked very hard to forget a lot of this stuff, but I'll try to remember what I can to help you guys out. So if you go through DNA replication, you end up with 2 chains. They go to 2 different cells. The cell splits. If you go through transcription, you open up a site, and then a piece of RNA is produced. I'll use my thumb. This is a little bit different. A piece of RNA is produced, and eventually that piece of RNA goes out of the nucleus and is used to make proteins through translation. So at no point should the DNA be getting changed. So there's this idea that there are mutations in DNA, and there are features, and it is changing, but that's not within an organism. So an organism isn't usually undergoing mutation. Usually what happens is maybe mutation enters a population. So it does start with maybe one organism, but it doesn't happen usually in their lifetime. It's not like they get irradiated and suddenly they're the Incredible Hulk chicken or something. But they have something that gives them a survival advantage, and so those genes will get passed off to more offspring. So it's not like magically all these chickens mutate. It's just a shift in the population over time. If it's advantageous. If it's not, if it's a disadvantage, it should go away or become less common. And it should become more common if it's favorable. So we want to make sure we keep this data fidelity. So the first thing is, if you've got, you essentially have 2 copies right off the bat. You got one strand, you got the other strand. So you have a complementary strand that goes with it. And so if there's anything mismatched between those 2 strands, there's a enzyme that runs up and down the DNA, and it just looks for errors. And if it sees that there's a mismatch, it fixes them. And if it's a mismatch, there's a lot of different options. Sometimes it's 50-50 on if it gets fixed right or not. Sometimes it's pretty much 100%. Sometimes the fix doesn't get caught in time before replication happens, and then you have 2 cells with those issues. And so one of them will have the correct DNA, the other one will have the mismatch that now is part of its DNA. And that can get passed off to offspring or something like that if it ends up being a sex cell. So there's a lot of stuff that can happen here. And we're just going to boil it down to, we're looking at the chemistry, we're looking at the molecule. How do we prevent that mismatching? How is it going to make sure that when I do replication, I have 2 matching sets of DNA? Well, if I have a G and I pair it with a T, look what we have up here. We have a hydrogen bond receiver. Look what we have up here. We have a hydrogen bond receiver. And then we have a hydrogen bond sender in the middle. We have a hydrogen bond sender in the middle. So they just don't match up. So it's going to try to receive a hydrogen bond on top, but they're trying to receive a hydrogen bond on top. Now when you have a correct pairing, it's trying to receive, and it sends, and so you get a hydrogen bond. But if you have two of them trying to receive, there's no hydrogen bond, there's no attraction. They're not going to sit right. They're not going to interact with each other. And so the enzyme will see that and it'll try to fix it. There'll be all these, things that will show up physically in the DNA to trip it off to that. And this is the first one. Now, if I try to pair these two, I get that issue. If I try to pair these two, I get the same issue. I get a hydrogen bond mismatch. So this is going to try to make 3 hydrogen bonds. This is only going to try to make 2, and they're opposite of each other. So over here, we got a send in the middle and a receive on top. And up here, we have a send and then a receive. So hydrogen bond is being received this way, hydrogen bond is being received this way. So if that seems weird, just remember this can't make a hydrogen bond, it can only accept one. So these are sending, these are receiving. And the same deal here, if I have a nitrogen with no hydrogen on it, it receives, it does not send. So it's a very key idea here. The other thing is, there's a pattern here. I've got a 2-member ring, 5-member ring. So I got 2 rings, a 5-member ring, and a 6-member ring. Sorry about that. 5-member ring, 6-member ring. That's all one molecule. This is all covalently bonded. So this is one big molecule. And over here I've got a 6-member ring. So it's about half the size. And over here I have a 6-member ring. And so what I have is I always have a setup that's like 2 rings, 1 ring. And so they're a certain size. So if I try to pair guanine and adenine, they're too big. It should be like this. This is too big. That's an issue. If I try to pair these, they're too small. So even though we could potentially get the hydrogen bonding to maybe match up, the enzyme's going to catch it because it's not spaced correctly. So everything, whether it's like this or like this, it will always be in those 2 strands a certain distance. And if I mismatch, basically up down here, I get something that's too small or something that's too big. So size plays a factor as well as hydrogen bonding. So just be aware of that. So that is how errors are detected in DNA. And so again, don't memorize the structures, and just remember that hydrogen bond pairing holds them together, and it holds the 2 complementary strands of DNA together, and it helps prevent error corruption, basically, as well as the size. So there's a lot of cool kind of chemical features built in. And so these are all the pairings you'll see in DNA. Now, when we're working with RNA, generally it's a single strand. We'll talk more about that later. But when we are doing transcription, you still get a pairing because you've got to pair an RNA nucleotide to a DNA pair. And then it just peels off and the DNA closes back up. But anyway, we get an AU pair. So there is no thymine in RNA. There is what's called uracil. And let's see if I can line this up right. So we'll compare thymine here and uracil really quick. They're very, very similar. On the left-hand side, they are identical. On the right, thymine has 1 more carbon. We call this a methyl group. It has 1 more carbon. Whereas, and then the double bonds shift a little bit. But so the right-hand side gets a little bit different, but the left-hand side here is identical. So they're very similar. So what it does is, it's a 6-member ring, so it's the same size. And 2, it has the same hydrogen bonding pattern. So it receives up high, and it sends kind of in the middle here. So it'll send a hydrogen bond, and it will receive a hydrogen bond in the same locations that thymine did. And so that allows the pairing. But you will only see uracil in RNA. So this is RNA only. So it's one of the first different, I guess it's actually the first difference I've told you about DNA and RNA. Other than the DNA is double-stranded, RNA is always single-stranded. It's kind of, it's an open-faced sandwich. DNA is a closed sandwich. We don't care, whatever. We drop the open-faced sandwich and it lands butter side down on the ground. We don't need to eat it. We'll just make another one. We know how. We got the recipe over here. Nice and safe. So that's what we do. So these are all of the nucleic acids. And again, you don't need to memorize the structures, just those key features I talked about. Make sure you understand that. Make sure you understand the role that hydrogen bonding is playing here, because these are molecules that are interacting with each other. So attractive forces are incredibly important. They're driving all of this. So there is no covalent bond between DNA strands. Within a strand, it's covalently bonded, but that complementary strand is not covalently bonded to the original strand. So I have two large molecules that are covalently bonded, and then they attach to each other through a ton of hydrogen bonding. That's what's going on. And then in RNA, it's single-stranded, but it needs these hydrogen bond interactions to produce the RNA chain off of the DNA. So what happens is the transcription process reads the DNA one base at a time. If it sees an adenine, it throws the complementary uracil into the RNA chain. That's what happens. And then that RNA chain gets built one piece at a time, and when it's done, it gets sent off to be the blueprint for proteins. All right, so this is the introduction to the nucleic acids. So we talk about all the structures here. Normally, I like to, write it out as I go. I think you might recognize why I chose to draw these in advance. I have at least one sheet of paper on the floor already where I screwed stuff up. And like this is a nitrogen, just to be clear. It was a little bit of a heartbreaker to almost screw up writing an N. I went this way, so it was like right at the end. It was literally at the end. And then this one, I started to misspell uracil, and I was like, I'll just rewrite it, whatever. So, it's not like I didn't make mistakes still, but they were small ones. So we're going to talk more about this backbone piece in the next video. We're going to talk about how these connect to them, and then how these long DNA and RNA chains are made. So I talked a lot about what holds them together, what makes sure that they are put together correctly, that I have what I have over here and over here correctly line up with each other. So if I have, a G, do I have a C in the DNA? And then how does that apply to making an RNA chain? I talked about a little bit. A lot of that is just kind of to give you a leg up in the biology class though. So don't panic about it too much. Mostly it's no nucleic acids when you see them, know their function. And their function is, if it's DNA, long-term genetic information storage should not be getting changed. Imagine a file that you do not want getting corrupted on your hard drive. That's DNA. How do we prevent it from getting corrupted? Well, you check it for errors constantly. Now they're big molecules, and so there's a protein that just has to zip up and down the chain looking for errors. And so it takes time. So it's possible for an error to occur, and before the checking protein can get to it, you've already had DNA replication. Now you have a cell with an error. So sometimes people that are high risk for cancer because of certain genetic factors, one of them can be the efficacy of that error checking protein and how frequently it's happening. So that's not a set rate among all people. That's something that can vary, and it can vary from tissue to tissue as well. So there's a lot in play there. All right, so we'll call it here. This video is getting a little bit long. So next, like I said, we're going to talk about building these up. So we're starting. This is small, believe it or not. Like these feel like they are massive molecules, but these are the small pieces of nucleic acids. So we're going to build them bigger and bigger and bigger. All right.